wanna dance by the water neath the Mexican sky. Drink some margaritas by a string of blue lights. Listen to the mariachi play at midnight. Are you with me? Are you with me?
Okay, good morning. I'm sorry to disappoint you, but Pozueta is not here today, so I'm gonna take over. So, as I said the last time, I think probably I will be quicker than Javi. First of all, I don't have any ephemeride prepared, but today is Carmen's birthday, so let's wish Carmen a happy birthday. Well, we can sing it as well if you want. Año feliz, cumpleaños feliz, que sea todo, cumpleaños feliz. ¡Bravo! Okay, cool. Any other birthday today? No? Okay, well, it's a pleasure for me to introduce you Claude Becker. He's a good friend of us, Pablo, Carmen, and me. So we met at the Max Planck. So I'm going to tell you briefly some of his biography. So he's a biologist. He studied in Heidelberg, right, the University of Heidelberg. And then he did a PhD at the University of Freiburg, where he graduated in 2010, if I'm not wrong. Then he moved to Max Planck, where, as I said, where we met, and we coincided for around 3 years. And it was a really fun time, I have to say, with this guy. You will see, he's an amazing scientist, but he's a really bad poker player, I have to say. He doesn't know how to cheat. He's too honest. He's too German. No, he's not German, but actually, but Germanized. So then he moved to GMI, to Gregor Mendel Institute in Vienna in 2016, where he started his own group, and he started his own research line that is related to what we are going to see today. Is related to the interaction between plants and environment, and specialized metabolites and genes that are involved in the synthesis of these compounds, mainly, I guess, microorganisms. No? Then he moved in 2019 to LMU, it's Ludwig Maximilians University in Munich, where he's now the associate professor. Has his group there. Well, he got a ERC as well. He's starting grant. Just it's something important to say. Yeah. Well, I got it, so I have to also say that it's important. So, well, that's more or less. It's an amazing track of publications, and so I hope you enjoy the talk. And yeah, it's your turn.
All right. Good morning, everybody. So after singing, at least everybody's awake. So thank you very much for the invitation. This is my second time to Malaga, but my first time to this institute. And after Pablo made fun of me for providing a very generic title 6 months ago, I decided to change the title and to be a bit more specific about what we actually do. So the title is What's Your Toxin?, which also relates very well to yesterday's evening with these guys in the city center. And it's about how plants use specialized metabolites to shape the living environment that surrounds them. So the US is the only place where you can find numbers on this, but every year in the US, health centers report about 100,000 cases of people reporting some plant-related poisoning because of inadvertent ingestion of plants. So this is excluding, of course, everything related to tobacco and all sorts of drugs that people take deliberately. So this is inadvertent poisoning. Fortunately, very few of these are lethal because there's very few plants that are absolutely deadly, but they exist. So I've picked some examples here. If you watched Breaking Bad a couple of years ago, you will remember this one here, castor bean, which is producing ricin, which is actually a peptide or protein that is a ribosome inactivating. And it takes about 8 beans to kill an adult. More extreme are rosary peas, which produce a very similar protein, also inhibiting ribosome activity. And it takes only 1 of these seeds to kill an adult person. So these are protein toxins, but many toxins in plants are actually specialized metabolites, which until recently were cataloged as secondary metabolites to distinguish them from primary metabolism. But because secondary is worse than primary, so people now refer to them as specialized metabolites. One of the deadliest specialized metabolites in plants that you can find is cyclotoxin from water hemlock, which is the one that's causing most cases of death in the US, particularly in children who mistake this for some carrot plant because they look very similar. And of course, everybody knows deadly nightshade, belladonna, which is producing atropine, an alkaloid, and 6 berries of these are sufficient to kill an adult human being. So what I'm trying to illustrate here is that plants are master chemists, and current estimates run as high as up to 2 million different compounds that plants are able to produce across all species. This is an estimate, so most of these compounds currently remain to be identified, and we don't know what they actually do. But many of these are biologically active, which means that they influence other organisms, and this is important if you consider that plants are hubs of organismic interactions in both the beneficial and the pathogenic sense. So plants interact with insects, be it as pollinators or herbivores, with viruses, with all sorts of fungi, beneficial and pathogenic ones, bacteria, and so on. And so many of these interactions are modulated by specialized metabolites that are produced by the plant and very often also released into the environment. Okay, so when I set up my lab at the GMI, we started to focus on a very particular type of interaction, which was plant-plant interaction, which we realized quite rapidly also involved plant microbe interactions. And so the particular scenario that we started to focus on initially is this one here, which ecologists refer to as allelopathy. It involves a donor plant that produces and releases specialized metabolites, which in this case are referred to as allelochemicals. And these chemicals will enter the soil, either directly from the roots or from rainwash or leaf debris or whatever. So they end up in soil, and they will affect the growth and the development of neighboring plants. And this can happen directly, or in some cases, these plant-derived compounds need to be processed by microorganisms to convert them into the actual phytotoxin. So this is a phenomenon that has been observed centuries ago. If you buy gardening books, they will tell you what not to plant next to some plant growing in your garden because it will inhibit the growth. And also the first systematic experiments were done quite a while ago. So this is a photograph taken in 1900 in a British orchard research station by this gentleman here. And so what these people did is they planted apple trees. These are clonally because they are grafted. So these trees on the picture are all the same age. They were planted at the same time. They are clones of each other, but you can see that they are markedly distinct on the left and the right side, and the only difference here is that on this side of the garden plot, people left the grass to grow, while on this side they kept constantly removing it. And so we now know that grasses are very good at producing specialized metabolites that are growth inhibiting to other plants. And we also know that apple trees tend to be very sensitive to some of these compounds. Another historic example is this one here, which now is more than 100 years old. So this is the top view of a field plot. There's a walnut tree growing in this upper left corner here. This is the crown of the tree, the limit. And then people started planting tomato plants in increasing distance from this tree, and they noticed that these plants were either very sick or even died up to a certain point, after which they looked healthy. And after they started digging, they'd realized that this demarcation line coincided exactly with the limits of the roots of this walnut tree. And so we now know that walnut trees produce this fairly simple compound here called juglone, and that juglone is very potent in inhibiting solanaceae plants such as tomato. This is just a fairly random assortment of different crops that are producing very different specialized metabolites, just to illustrate that when we are talking about growth inhibiting substances, these can be chemically very diverse. So we have this very simple molecule, juglone here, I'll talk more about momilactin later on. But there's also things like this, sorgoleron, which is basically an oil, if you want. So if you grow sorghum plants on a petri dish, you can see dark brown droplets forming on their root hairs, which consist almost exclusively of sorgoleron. Right, so what we are studying in my lab is 3 different components of this. So first of all, we would like to understand how plant-derived metabolites affect other plants. So basically, what's their mode of action? We also want to understand how the pathways have evolved that produce these secondary metabolites and how these pathways are regulated. And ultimately, we're looking into the role of the microbiome that's surrounding the plant in modulating these chemical interactions. So we do this by using 3 systems, so to speak. So the first one is several grasses that produce benzoxazinoids. So that's this compound here. You might have heard of DBOA if you're into herbivore repellent agents in plants. We work on momilactones, which are produced in rice, and we work on withanolides, which are produced in Solanaceae species. So these are, chemically speaking, these are very different compounds. But I'll show you one similarity, and that is that all of these are produced by genes that are encoded in so-called biosynthetic gene clusters. So biosynthetic gene clusters are prominent in plants and in fungi, but they're very rare in animals. There's very few examples of animals having a biosynthetic gene cluster. So what are they? They're basically genomic regions in which you have a co-localization of heterologous genes, which means they have different origins. They're not just simply paralogs of each other, but they have different origins. But they've aggregated in a genomic locus. And they've done so because they encode enzymes that catalyze the subsequent steps of a biosynthetic pathway. By being aggregated in this genomic locus, they can be transcriptionally co-regulated very easily, and it ensures their co-inheritance. So the pathways don't break through recombination leading to some toxic intermediate. You will always have the whole pathway inherited. And they are, it turns out, a core feature of evolutionary innovations in plant-specialized metabolism. So when we talk about evolution, and of course we have some processes that are very fast evolving, like this one here, for example, and we have others that evolve very, very slowly, if at all, like this one here. So plant-specialized metabolites definitely belong to this upper category. It's a very dynamic evolutionary process that drives the diversification of these metabolites. So I'd like to tell you 3 stories related to these 3 study systems that I mentioned. And I'll start with the benzoxazinoids. So these are multifunctional regulators of organismic interactions. They have been known as herbivore repellents for decades. They are now also known as iron chelators that the plants exude into the soil and then take up again after they have bound iron. But they also inhibit the growth of other plants and bacteria. So I mentioned to you that all of these compounds are originate from biosynthetic gene clusters. So this is the cluster in maize on chromosome 4. And it contains not all, but most of the enzymes that are necessary to produce benzoxazinoids from indole glycerol phosphate. So it takes 5 consecutive enzymatic steps to produce the active compound, DBOA here. And then in the leaf, the compound, just to prevent autotoxicity, will be stored as a glucoside. The glucosidase is stored in the chloroplast, and when the caterpillar comes and chews into the leaf, then these 2 things come together, the sugar group is cleaved off, and the active form is entering the gut of the caterpillar and inhibiting digestion. Okay, so another thing that seems to be recurring in specialized metabolism is conversion evolution. So benzoxazinoid biosynthesis emerged several times in land plants. So I told you they're produced in grasses, which is up here, but they've also been detected in some very distantly related eudicots. So this and the biochemical steps are very similar, but the origin of the biosynthesis is independent. Okay, so once DBOA exits the root into the soil, this compound is very unstable. So within hours or days, it will be converted to this intermediate compound here. And then, with the help of microbial activity, in the end, it will result in this compound here, aminophenoxycidin, or APO for short. Now, APO, in contrary to the precursor, is very stable. You can detect it for months in soil, and it is also the biologically more active form. So if we compare plant growth inhibition using either BOA, which is this intermediate here, or APO, which is the final product, there is basically more than 100-fold higher inhibition activity by APO, if you look at Arabidopsis root growth, for example, shown here. So how does this work? Well, we still don't know for sure, but we found one mechanism on how APO acts on plants. And this was from a project that originated, strangely enough, in the university clinics in Tübingen, and then we followed up on this. So I have to make a short excursion here into chromatin regulation. So in case you're not familiar with this, histones can be modified at their N-terminal tail, and depending on the type of modification, this will affect the structure of the chromatin. So histone acetylation is one of these marks that will, when attached to the histone tails, will open up the chromatin. The enzymes that attach these acetyl groups are histone acetyltransferases, and those that remove them are called histone deacetylases, or HDAs. So there is a couple of well-known HDA inhibitors that are also used in cancer treatment, for example. One of them is SAHA, the other one is trichostatin A. And we know where these compounds bind to the HDA catalytic site. And so with the help of docking simulation, we could show that these plant-derived compounds, the stable ones that originate at the end of the cascade, can bind to the very same pocket. And if you treat Arabidopsis plants with either one of these, you will get a global increase in H3 acetylation. Particularly, you get an increase in H3 K27 acetylation for the aficionados among you, which means that this affects gene regulation, but it potentially also affects cell function, cell division, and so on. So this is one mode of action of APO, but we suspect that there are more that ultimately lead to this growth inhibition. So this was some time ago, and then we noticed in the meantime that APO not only inhibits plant growth, but also lots of bacteria that are associated with the root. And so we asked, what are the roles of these benzoxazinoids in altering the soil microbiome, and how would this affect plants that grow then later on in that soil? So there was a very interesting study that was published by our collaborators from Switzerland, where they did the following. So they grew maize plants. So maize is a benzoxazinoid producer in soil in large pots for several months. And they grew two types of maize: wild-type maize, which produces benzoxazinoids, and a mutant interrupted in the biosynthesis. So this is a normal-looking plant, but it doesn't produce these compounds. They took out the plants, but they kept the soil. And they put a second generation of maize into that soil. And they noticed that when they did that in the BX plus soil, so the soil coming from the wild-type maize, plants would be smaller, but they would be more resistant against herbivores and pathogens compared to plants growing in this BX minus soil. However, when they sterilized the soil in between, the effect was gone. This means that what they observed here is not a chemical effect per se, but it's a lasting effect on the microbiome. So the chemicals modifying the microbiome, and the microbiome feeding back on plant growth. They could replicate this by just adding the chemical. This would lead to the same alteration of the microbiome, which would lead to the same feedback on the plant. Okay, so we wanted to understand, so just to recap. So benzoxazinoids are released by the plant. They change, or we call it condition, the soil microbiome, and this conditioned microbiome produces a feedback on the next generation of plants. So we wanted to understand what the genetic basis was on the plant side in responding to this altered microbiome. And because we can't do this very well in maize, we did it in Arabidopsis. So we asked our colleagues in Switzerland to repeat the experiment. So they grew a lot of maize in large quantities of soil, and then on a weekend, when Swiss customs was asleep, we shipped 2 tons of soil from Switzerland to Vienna, and set up an experiment in our phenotyping chamber. So we grew 400 Arabidopsis genotypes in both soil types, and we phenotyped them for several weeks, which led to 300,000 images. In case anybody here is interested in extracting phenotypes from your plant data, I'm going to do a little parentheses here, just telling you about a tool that we developed specifically for this project. So if you try to phenotype plants, it all works well as long as your plants look like you know them from your greenhouse experiment, right? If you grow Arabidopsis in field soil like we did, they will look like this. And your phenotyping, the regular phenotyping, which is colorimetric based, will fail. And so we came up with a machine learning based tool that is able to not only recognize this as a plant, but even catalogs healthy looking leaves from anthocyanin rich leaves to senescent leaves and so on. And so we applied this to all our 300,000 images, so you can extract all sorts of different phenotypes using this tool, that size and color and so on. So just in case this is serving your needs, you can look this up. Okay, so when we did this, we saw things like this. So we saw some genotypes that would prefer to grow in this BX minus soil over BX plus soil. But we also saw Arabidopsis genotypes that did the opposite. So they would prefer growing in BX plus soil over BX minus soil. And so we did this for all 400 genotypes, and we got a phenotype spectrum. Colombia, which is here, slightly favors growing in BX plus, but it's a relatively weak effect. But you can get extreme results in this distribution. I'll show you one here. So this is one of the extreme ones. This is the same genotype grown at the same time. The only difference is the history of the soil. 2 different maize genotypes had grown in that soil beforehand. And so you can see that there is a drastic difference between how these plants perform, depending on the soil history. So now we had this phenotyping over several weeks for 400 genotypes. So now we could do a genome-wide association study. But because this was a time, phenotyping over time, we had to look for GWAS peaks that would be consistent over periods of the experiment. So basically this is, imagine you're looking top down on a Manhattan plot and you only see the peak sticking out of the clouds as colored dots here, and everything else is hidden under the white layer. And you have a time axis here on the Y. So when you see dots that form lines, this means that this peak was consistent over several days or weeks even. And so we focused on these, using growth, just for a start, just relative growth as a phenotype. And so we had a couple of significant peaks here. I'm only going to talk about this one here because that's the one that we followed up so far on chromosome 3. So when we look at alternative and reference allele distributions at this particular locus, we see that plants, genotypes carrying the alternative allele perform differently on this, so they tend to grow better on BX plus soil, which is the blue one here, than those with the reference allele. It's not too many, the allele frequency is quite low, but it is significant. And the black bars here indicate those accessions that carry this alternative allele. So we then did an overlap of our GWAS peaks with a transcriptional analysis. So we did an RNA-seq on plants growing in BX plus soil and BX minus soil, and we checked, well, are any of the peaks from GWAS overlapping with genes that are differentially expressed? And there were exactly 3. And one of them is overlapping with the peak that I just showed you on chromosome 3, and it's in a locus called dangerous mix 2. David might remember this from and Pablo from Detlef's lab. So dangerous mix 2 is this one here. Our SNP is next to the neighboring gene. So it's an NLR, which means it's an internal effector receptor, and we called it mediated to microbiome feedback 1, MMF1, because it didn't have a name before. So in Colombia, I told you there was a weak but significant positive effect of BX plus soil, which is shown here. If we knock out MMF1, which is the one over here, this is a knockdown, this is a knockout. If we knock out MMF1, we lose this soil response, so it's gone. We then did a transcriptome profiling of the wild type and the MMF1 mutant in both soil types. So what you can see is that there is a distinct transcriptional response to soil type in the wild type, which are the dots here, so the green dots separate from the yellow dots, but not in the mutant, which are the triangle. So the triangle is blind to the soil history. We also looked at the microbiome, and so first of all we see that BX plus and BX minus soil leads to different microbiomes, which is essentially the yellow separating from the green. But we also see that the root microbiome differs between wild type and the mutant only in BX plus soil, but not in BX minus soil. So there is a clear effect of losing this one receptor in responding to this conditioned microbiome. We still don't know what the effector is, but this is what's happening. So when we look at the locus, you might have noticed that the SNP that we score is not in the gene. It's somewhere outside the gene. So what was known already about this locus is that it's highly structurally diverse among Arabidopsis genotypes. And so to figure out if this was maybe hiding a larger structural variation in this locus, we took some of the extreme accessions from our phenotype distributions and we did long-read sequencing to assemble their genomes. So we took 20 of them, we assembled them, and then we basically scored structural variation across the genome. So the higher the peak, the more structural. This is basically a genome graph. It's a summary of a genome graph. The higher the peak, the higher the structural variation at this particular locus. So you get a lot of this in the pericentromeric regions, which is expected. But we also got one of these very distinct peaks exactly overlapping with our locus of interest. So there's a lot going on there, which is illustrated here. So this is basically this one locus across these 20 accessions. And we see everything you can imagine. We see 5 copies of this NLR, we see 1 copy of the NLR, we see additional ones, we see loss, and nothing of it makes any sense. So we see no pattern. This is ranked along the phenotype distribution. There is no pattern in the structural variation. But there is a pattern when we look at the protein domains of this NLR. So there is a nucleotide, there's an NBS domain here that shows a distinct change between plants growing well on BX plus versus those that don't. And there is another one which is a predicted nuclear localization signal that exists only in a fraction of them, but not in the other. So these are the 2 things that we think are responsible for this. So we are now following this up with doing allele swaps between genotypes and generating chimeras of these NLRs. The other thing we're trying to do is we are brute forcing AlphaFold 3. So we're basically running all the known NLRs against all the known effectors, which is thousands and thousands of combinations, to find those effectors that are predicted to be able to bind to our specific NLR, hoping that we can narrow down to a list of, I don't know, let's say 10 to 20 candidates that we can then genetically follow up on. All right, so to summarize this part, so I've shown you that benzoxazinoids are multifunctional compounds that mediate a lot of organismic interactions. They are converted in soil by microbial activity, and then they turn into these phytotoxic compounds. They are long-lived, and they lastingly alter the root microbiome or the soil microbiome, and this in turn feeds back on plant growth. Okay, so now moving on to the different class of compounds, which are rice diterpenes. So these are essentially inducible chemical defenses against neighboring plants, but also against pathogenic fungi. So this is what they look like. They're rather complex molecules. There's 2 forms, A and B, that have been characterized to some extent, and then there is minor forms, C, D, and E, that people have detected, but nobody has done anything on them because they exist only in tiny amounts. As I said, they are growth inhibiting, so you can very efficiently inhibit Arabidopsis growth, and they've also been shown to inhibit, for example, Magnaporte colonization of the plants. So studying the biosynthetic gene cluster of this was part of Santi's PhD thesis. So this is the enzymatic cascade here, which is only part of it. And this is the cluster in rice. So Santi actually did a phylogenomic comparison to see how this cluster had diversified within the Oryza genus. So not just in cultivated rice, but also in the wild relatives. And he could show that, well, this cluster likely originated from an ancient gene cluster, which is also which exists in other grasses, but produces other types of diterpenes there. So, for example, there's homologies to a functionally distinct cluster in the Triticid tribe that also produces diterpenes, but they are different from momilactones. And this cluster had diversified in the Oryza genus. So Santi found events of loss of this whole cluster, but also slightly different architectures that he could use to retrace the phylogenomic history of this. So this was looking across the Oryza genus. So currently we're looking across Oryza sativa in particular. So we're making use of the 3,000 rice genomes resource. As the name says, there's 3,000 cultivars or land races that have been fully genome sequenced. Santi, before he left, did us the favor of building this core set that we could use of 360 accessions that represent most of the genetic diversity there. And so we measured momilactone levels across all of these 360, and what we see is a 10,000-fold difference in concentration between different cultivars. So we still cannot explain this. We are now mapping this back to the genetic data, but what we can see and what others have also observed in the past is that Japonica rice, which is this one here, has on average much higher momilactone concentrations than indica rice, for example. Okay, so momilactones have been characterized as something that exists in rice. So almost all the papers on momilactones are on rice. There's one on another grass, Echinochlora, and then there is one case in which momilactones have been detected in mosses. So that's a bit weird, right? And this comes back to this concept of convergent evolution, which I already mentioned. So convergent evolution is you get the same phenotype in different ways, right? And so this has happened. Yeah, I couldn't resist. So this is what's happened to the momilactone cluster as well. So this is the biosynthesis in rice from the cluster that I just showed you. And this is the biosynthesis in moss, which uses different enzymes from an evolutionary standpoint. They have different origins. But strangely enough, they also aggregated in a cluster in moss. So Peter, a PhD student in the lab, asked whether this one discovery in moss was a fluke or whether this was maybe a more common feature in mosses. And so he looked at the moss phylogeny, which is a very complex one. There's many moss species, and they all look the same to me. But he teamed up, so this is the cluster in mosses. So he teamed up with a bunch of botanical garden curators and bryologists to get moss collections from all over Europe, from Scandinavia, from Scotland, from Germany. And then we ran mass spec on these, and he was able to detect momilactones in 11 new species that had not been described before. And they distributed across this bottom clade here. So this is basically the minimal common node that we could identify. This seems to be a very widespread phenomenon in mosses. Just because we could, from the Munich herbaria collection, we also took some mosses from there, and we asked, well, can we actually detect momilactones in historic samples from herbaria? We didn't expect anything to come back from this, but just for fun, as we were running the samples anyway. And indeed, we were able to detect momilactones in samples that were 100 years old or older. So there seems to be a point where it starts to deteriorate at some point, and it also, you can see it in the bottom here, it seems to depend on how the samples were handled in the herbaria, but it's technically possible to detect these compounds over long time spans. So we also went and for 4 of these or 5 of these mosses, we assembled their genomes just to see if the cluster would be conserved as well. And indeed, so when we look at the cluster across these newly assembled, this is the one that was already known. So we checked against 4 others, and we could find the exact same cluster in 3 rather distantly related species. This one here, even though we could detect the compound, we couldn't find any of the genes so this is still a bit of a riddle. Currently, Peter is looking into yet another completely different plant family, which are tropical trees. There was one paper that indicated that a momilactin-like compound existed in tropical trees, and indeed, again, touring some botanical gardens in Germany and Italy, he collected a bunch of species from these families, and he was able to find some that unambiguously produce momilactin, at least one form of it. And so this might be yet another convergent evolution event. So momilactins are diterpenes that have antifungal properties, but also growth suppressive properties. What I haven't shown you here is that they are also required in rice to induce arbuscular mycorrhiza, it seems. The production varies substantially among rice genotypes, and there are several, at least two, possibly more convergent evolution events that lead to their biosynthesis. And as it turns out, mosses might actually be the overlooked and most promiscuous momilactin producers around. Okay, which brings me to the last story I wanted to tell, which is about withanolides. So these are, again, chemically speaking, very different compounds. If you are into nutrient supplements, you might have one or the other ashwagandha product at home that you put into your smoothie or take as a pill. And so part of the active ingredients in ashwagandha is this one here, withaferin A. So ashwagandha, if you don't know it, is the winter cherry, Withania somnifera, hence the name of the compounds. So this is a project that was started initially by Eva Knoch, used to be a postdoc in the lab, and then taken over by Santi. And the interest in withanolides is because they have all these supposedly beneficial properties. So they are anti-inflammatory. There's been a Nature Methods paper last year showing that they can suppress leukemia. They have antibacterial properties, at least against some strains. And in plants, of course, they are, as most specialized metabolites are, defense agents. In this case, they repel herbivores, at least that's the suspected function. So again, they're pretty promiscuous across the Solanaceae. There's different branches of this family that have been shown to produce withanolides, and they're chemically hugely diverse. So up to now, there's 600 different forms of withanolides that have been characterized in only a few species. So this biosynthesis branches off that of brastinosteroids, and this is the first dedicated step catalyzed by this delta-24 isomerase, which converts this compound here into this compound here, and then it continues to withanolide biosynthesis. So when we started on working on this, or Eva started working on this, because we are a genomics-oriented lab, we wanted to work with something where we had a genome available, which is not the case for ashwagandha, at least it wasn't back then. And so we reverted to the most closely related one, which is physalis. So you might know these from your ice cream cup. So they are closely related to ashwagandha, and they produce similar compounds called physalins in this case, but they are, chemically speaking, they're the same thing. And so the first thing Eva did was she looked for her gene of interest, this 24 isomerase in the physalis genome, and she noticed that there wasn't one copy, there was two copies, and they were sitting in fairly close proximity to each other. And surrounding these two copies were other genes that looked as if they could be part of a biosynthesis pathway. Curiously, not only the 24 iso existed in two copies, but many of the other genes also appeared twice in this genomic locus. So Santi carried out a phylogenomic comparison, so don't worry, you don't have to follow all of this. What I just wanted to highlight here is, so physalis is up here. So we have this cluster which shows this apparent duplication of many of the genes. And so by comparing clusters across species, Santi could figure out that there had been a duplication event that must have happened somewhere around here. Not in any of these species, and then there was a second duplication event, independent presumably, that happened in this Datura clade down here. You'll also notice that most of the Solanum species listed here, including tomato and potato, do not have the cluster and do not produce withanolides. So this seems to be a dichotomy. Plants either produce steroidal glycoalkaloids, like potato, or they produce withanolides, but never the same, never both of them at the same time. Okay, so the first thing we did was to look at the transcription profile of these genes, and we sampled different tissues of Physalis plants, and lo and behold, when we looked at the expression profile, we saw that the first part of this cluster, or the first set of genes here, was expressed exclusively in roots, whereas the second part of the cluster was expressed predominantly, not exclusively, but predominantly in aerial tissues. So we called this the root-expressed subcluster and the shoot-expressed subcluster. We also, because we'd seen that this duplication had originated here, we wanted to see whether this behavior was the same in other species. So Santi again took a trip to the botanical garden and sampled some of these plants. And indeed, we see very similar tissue-specific expression patterns in tuber capsicum and in Euchroma, which phenotypically are very different species from Physalis, as you can tell, but this seems to be conserved. So together with Alistair Fernie at the Max Planck, we also looked at the metabolic profile, and we could see that withanolide compounds, which they call features, are also very tissue-specific. So we could find some features that existed only in roots, only in flowers, only in leaves. So basically every tissue had its specific class of withanolides. Okay, so but is this supposed cluster really contributing to withanolide biosynthesis? So at the time we were not eager to do a lot of knockouts in Physalis because that takes time, but you can do VIGs in Physalis, so virus-induced gene silencing. So if I tried this, so she knocked out a couple of the genes from this shoot subcluster via in leaf infiltration assays, and we could show that by doing this, the withanolide content would actually drop. So these are not knockouts, these are knockdowns, but it's sufficient to drive down the biosynthesis. So in the meantime, two other labs have studied the very same cluster in ashwagandha, and they could unambiguously show that this cluster is the one that produces withanolides. Okay, we were more interested in how this whole cluster subdivision would work at the molecular level, because this was on a very short distance, you get this very distinct behavior, differential behavior. So the first thing we did was to look at chromatin-chromatin interactions, so the chromatin architecture around these clusters. So we teamed up with Chang Liu to do Hi-C in Physalis. So this is the whole Physalis genome. The only thing I want to illustrate here is that so this is a 1.5 gigabase genome, and 90% of it is heterochromatic. So everything in green here is heterochromatin. And the genes are clinging to the chromosome ends on each chromosome. So our cluster of interest is located here, the end of chromosome 1. So when we zoom in to this euchromatin region, this is our cluster here. And you can see that on the Hi-C map, something's going on here. This is the distinct region from everything else in the euchromatin, which prompted us to look at higher resolution, and we could see that if you look at chromatin-chromatin interactions, there is a lot of interaction going on within this shoot-expressed subcluster, but there is basically no interaction between the shoot-expressed and the root-expressed subcluster, which would be lighting up here. So there's two chromatin domains that are separated from each other, it seems. Santi also did this in shoot tissue compared to leaf tissue. And if you look at the differential, then you can see that by this blue color, that the root-expressed subcluster is more condensed in the leaf, where it's not expressed, compared to the root. So it seems that transcriptional suppression, in this case, goes together with higher compaction, or more frequent interaction, at least, of this cluster, which is exactly what this says. So me having a background in epigenomics, before I ventured into the specialized metabolites, I was curious to see what the epigenetic landscape would look like in this cluster. So we looked at DNA methylation, and here again we could see a very distinct shift in DNA methylation between this root-expressed and the shoot-expressed subcluster on a very short distance. And we could see a very distinct shift in DNA methylation between this root-expressed and the shoot-expressed subcluster on a very short distance. And we could see a very distinct shift in DNA methylation between this root-expressed and the shoot-expressed subcluster on a very short distance. which was inverted depending on the type of methylation that you looked at. So this is symmetric DNA methylation, and this is small RNA dependent DNA methylation. And so they behave in opposite ways. We still don't know what this means, if it's just the collateral or if it's actually something causal. And then lastly, we also looked into local chromatin accessibility using ATAC-seq. And as we suspected, this root expressed subcluster is pretty much closed in leaves, but it's open in roots, and it's the other way around for this one here. So currently, we are working now with knockout lines. So we basically we deleted this whole cluster as a half a megabase. We just cut it out, which worked on the first try. And what didn't work for 2.5 years was cutting out the subsections of the cluster, but now we finally managed. So we cut out the root expressed one and the leaf expressed one. And we also cut out this gene-free spacer in between, because we suspect that this has some structural function as well. So just to give you a glimpse, I'm almost done, on what we are doing with these lines. So we teamed up with Astrid Roth at the University of Amsterdam. So she's the only person in Europe who is able to culture these two moths, Chloridea subflexa and Chloridea virescens. So they are sister species, but they have very different feeding behaviors. One of them is a specialist who likes to feed on physalis, particularly because they contain methanolites. And the other one is a generalist who avoids physalis because it can't tolerate methanolites. So we did some feeding assays. We gave them wild-type plants, and as suspected, the generalist didn't touch it. The specialist ate some of it. But when we knocked out the cluster, they both went ballistic on these plants. Similar things happen when you do necrotroph-fungal assays with botrytis. On the wild type, this is rather contained, and in the mutant, the lesions get very large, so the fungus spreads very rapidly. So this is not just an anti-herbivore compound, it's also an antifungal compound. It's not a general defense compound, it's acting against everything. So when we do Xanthomonas assays, they don't seem to care whether or not methanolites are present. So we're following this up with many, many more compounds, and with Astrid, we are now starting this project here where we want to understand what are the effects, not only on the plant, but also on the caterpillar, particularly on its microbiome. So you want to see what are the effects on the microbiome of the roots and of the microbiome of the caterpillar. Okay, so I've shown you that methanolites are highly diverse metabolites, particularly in Solanaceae. They are of medical relevance, which is why several labs have started working on them. They have antibiotic and anti-feeding properties. Their biosynthesis is governed by a biosynthetic gene cluster that is fairly complex and has undergone a duplication event. And if you lose it, then plants lose a lot of their defense capacity. So with that, I'm at the end. I'd like to advertise our campus near Munich. So if you have questions about the specifics, you can ask Santi, because he worked there for 4 years. So we are outside of Munich, just behind the city border, right next to this huge Max Planck campus here. This is the group, and it's not very recent state, but fairly enough. And then I'd like to thank all of the generous funders we've had over the time, our very efficient collaborators left and right, and again, you for your attention and for the invitation to Malaga. And I have been told that I should put this up so that people can scan the QR code. Thank you very much.
Okay, questions?
First of all, thank you for your presentation. It was magnificent. I would like to know if you think that the compounds that you have mentioned, like MMF1 or the BX compounds, could be used in biotechnology to have antibacterial properties or antifungal properties.
Mm-hmm. So MMF1 is a receptor, just to be clear. That's not a compound. But so the BX or any of the compounds I mentioned, repeatedly there has been this idea around that these could be used to produce new herbicides, new pesticides, you name it. So far, I haven't seen this come to fruition. I'm not sure if this is because of lack of trying or because there's other problems. So one thing to mention is that many of these compounds are not very specific in their targeting, right? So when you apply them, you don't apply them only to the organism that you want to get rid of, but this will affect many, many more at the same time. For example, the APO that I mentioned at the beginning, it was tested as an anti-cancer drug, and it killed the cancer cells, but it also killed the rabbit having the cancer cells. So, I guess it needs more investments from chemists to find the right modifications to these molecules to make them more specific.
Thank you.
Hello, Claudius. Pretty cool talk. I mean, I like especially the pictures where I was. I have a question regarding the first part and the last part. So I don't remember. The compound was named BXBR plus, right? BX plus.
So BX plus is what we refer, how we refer to the soil that contain. Yeah.
Right.
Uh-huh.
So the frequency, the allele frequency there was pretty low.
Yeah.
So do you know why? So because it seems to be an advantage. It has like a deleterious effect, or it's like localization effect. So it's only happening in accessions near some plants that produce it, or.
So we mapped it to the geographic map. There's no geographic clustering. The allele frequency might be low for several reasons. One, it could be just spurious drift. The other one is that it's as I've shown, it's a structurally highly diverse cluster, so we might actually lose a lot of the information in the sequencing data on this. So we would have to do long-read sequencing for all of them, which of course we won't. But it's something we're eagerly waiting for Magnus and Detlef to release the 1001 plus resource with all the newly assembled genomes, because then we could have a better glimpse of this thing. The SNP alone is also not this is not a causal slip because it's outside of the gene. So the locus is important, but the SNP probably isn't.
Okay. And second question, so when you show it the Santis clusters, so you're only showing the genes, so how you look at transposon level, because you work it quite some time with transposon as well, so I assume that you check it.
Yeah. Santi, you want to answer that? So I know that, so Santi did some TE annotation at the time, but for this wasn't as straightforward as it might be now. So there are lots of transposons in this. There's transposons everywhere in Physalis. It's really riddled with them. And they also are quite prominent in the cluster. You can see, maybe we can go back to this. So if you look in detail on the methylation map here, so the CHH methylation is not happening in the genes, right? So almost exclusively in the intergenic space. And there's tons of transposons there. One possibility is that this is a collateral of this chromatin being more open, right, so the whole machinery can get in and methylate, but it has no consequence. That's one possibility.
Okay, that's pretty cool.
Hello, nice talk, thank you. I have a question about BX way of action, at least in plants. If I got it right, you say that it inhibits histone deacetylase activity. And I was wondering whether you know or you have any idea of how the plant that produces the herbicide does not suffer, because I guess getting your histone deacetylase inhibited is not a good thing.
No, you're right. And so we looked into histone deacetylases, the sequences. We did alignments between the susceptible and the producer. There's no difference in the catalytic unit that would explain this. So there's several ways by which you can become tolerant or resistant to your own toxin. Either you have dosage compensation, you just have more gene copies. which for histone deacetylase is not likely. The simpler one is that you either export the toxin, you just bring it back outside constantly, or you modify it. And since the producer is adds a glucoside to the deboa anyway, when it produces it, it might also be able to just prevent toxicity by doing that. So that
Okay, and any idea how it works on the microbes?
So we did a similar quantitative genetics approach as we did for plants on microbes. We didn't really follow up on this, but one of the hits we got was, again, a deacetylase. Of course, not a histone deacetylase, because bacteria don't have histones, but a deacetylase nonetheless. Yeah, and then the person left, and then we didn't follow up on this, but the idea was that we would knock out this deacetylase in the strain, and then I think it was a strain that wasn't too easy to transform, because these are all root-associated bacteria. So they are not
You're a
that easy
plant
to work with.
biologist, I guess. I mean, I could not.
No, I mean,
imagine
we have
starting with-
microbiologists dealing with these things, but some strains we just can't get transformed.
Okay, thank you.
Thank you. Great talk. I was wondering, because while you have been showing that there are quite a lot of biosynthetic gene clusters that you have been checking basically in plants. Now we know that many of these BGCs are also being horizontally transferred to the microbiome or to other bacteria. Do you know, because you check basically only in plants, do you know or you have any idea if these BGCs that you have been checking are also transferred to bacteria? If so, what's the frequency?
So, to be a bit provocative, I think the frequency is 0, because somebody still has to prove to me that there is lateral gene transfer happening in plants. So a lot of this is correlative, but nobody's ever shown a causal.
Well, we know that there are a lot of plant hormones, for example, that are already in bacteria.
Yeah, but these are but this. So what you're saying here is that this would have to go from plant to bacteria back to plant, right?
Or only to bacteria, because basically the BGCs that you are working with are only in plants.
To the now I get it. Okay, so not back to another plant, not a plant to plant, but a plant to bacteria transfer.
Yeah.
I mean, these are eukaryotic clusters, right? They are huge. For a bacteria, this is a big thing. So I have a hard time imagining that an intron-containing eukaryotic cluster could work in bacteria. You would have to have so many modifications to this to function as an operon. I really don't see how that works.
Okay.
Because you need to change promoter sequences, get rid of introns. Like all of these things need to happen basically at the same time. So hard to imagine.
Good. Thank you.
The other way around might be easier, from bacteria to plant. This I can see happening. Single genes, yeah, but not a whole half a megabase, right?
I'm not sure if benzoxonoids are also produced by bacteria. I have to check. I should talk with Klaus about it. Maybe it's produced by...
So they convert them, so this for sure. So if they get in contact with the compound, some strains have the enzymes to modify them or maybe do one of the steps. I'm not aware of bacterial strains really producing them.
No.
It's possible, but I'm not aware of it.
No idea. I have to check as well. Thank you.
Hello. Here.
Where are we?
Thanks for your talk. And I have one curiosity, two curiosity, because my field of study was the study of the terpene synthase in fungi, in botrytis. So sometimes these biosynthetic gene cluster for example, you have 7 genes, but then you have like 40 different compounds. I mean, you have the first step of the cyclation in the representation that you show, for example, one protein, one compound, the next step, another protein, another compound. So sometimes, for example, this diterpene synthase can do different kinds of cyclation. Then the additional genes can like add the hydroxyl group in the different carbons on the structure. So it's the same in your case. I mean, do you have more compounds that are minority?
Yeah. So so let's maybe use this one as an example here. So this cluster does not explain the 600 different compounds that have been detected, right? There's a lot happening downstream of this cluster with modifying enzymes. And this seems to be a general theme, that very often you have the core biosynthesis organized in one of these clusters, and then the decorating enzymes are somewhere else in the genome.
And the second question is, have you tried or have you think about, because now regarding to the terpene synthase, there is like a new type of study that is use this biosynthetic gene cluster but take like mimetic precursor. So some modification to the geranyl pyrophosphate, for example, for your diterpene synthase cluster. So you get finally non-natural products with maybe better bioactivity properties.
Yeah, so we haven't done any of this, but we are currently reconstructing the whole pathway in an expression system, but we're not the first ones to do that. So Elizabeth Sattler did this years ago, where she took the whole momylactone cluster and put it into tobacco leaves and produced momylactone there. And if you have a system like this, then of course you can feed it different precursors as you suggested. Yeah, and then you might get different compounds. So our aim is mainly to look for how these minor ones, I can leave this up too, how these are made, the one at the bottom here. So they have been detected in mass spec, but nobody knows how they're made, and it's probably just 1 or 2 more branching points from the core biosynthesis.
Okay, thank you.
Hi.
Hey.
It was very nice talk. I was wondering, like, seeing that you have that many accessions and that you also did a lot of genomic analysis, I was wondering if you consider or did some pan-genomic or pan-transcriptomic analysis, because I think that you could have really like great insights about which structural variants correlates or are associated with a certain expression of the genes inside the locus. And also I think that, continue what Victor said, you also maybe can correlate the expression of certain genes or another genes to some compounds or minoritary compounds.
So you're referring to rice now with the different different cultivars. So there's more and more assembled genomes coming out, right? And the more there are, the better we can use them. When we started this, we only had the resequenced 3000. I haven't actually done an update on this in a while on the pan-genome availability of rice, but I'm sure there must be more. The question is whether this overlaps with any of the set that we have now used for our measurements. So we can't do the hundreds of mass spec measurements in.
I know it's
depending on how the panel changes, right? But yes, you're absolutely right.
Okay, thank you.
More questions? Okay, I will ask you. Regarding the first gene you were talking about, the MMF?
MMF1.
Yes.
Yes.
You said that it seems that it could be like a change somehow in the protein, no? Some domains that are missing is the hypothesis, no? Maybe.
Yeah.
But you also found differences in the expression, no? When you did the RNA-seq and when you found this one as a candidate, actually, right? or am I
Yes.
wrong? So my point is, when you check the sequence of the genome of this region, you didn't see nothing that was really, that you could correlate with these differential levels or expression. So my point is, I guess you don't know what's going on there, no? It's because it's also something related to expression, right?
So the expression that I showed was only in Colombia in response to the two soil types.
Ah, okay.
We didn't test all the different accessions for their expression
yeah.
level.
Okay.
Just
Okay,
because
I see.
we didn't want to do 400
Another question,
transcriptomes.
when you, in the last part, these mutants you got is in Physalis, I guess, no? Or,
Yeah.
The experiment you show with the, I think it was the fungi?
Yeah, the botrytis one, yeah.
But so you found this phenotype, no, in leaves, but which subset is the one that is missing there? Is the root?
So this was, sorry, going back to this. At the time we did this, we only had the full deletion, like the whole cluster.
Okay,
yeah.
because
we repeated it with the partial deletions. Let me see. If we only delete the root cluster, botrytis isn't affected. If we delete the shoot cluster, so there we had a fluke in the seed batch, so we had to discard. But I can tell you, deleting the root cluster, nothing changes for botrytis.
Okay. Any other? No? Okay, well, thank you very much.
Thank you.
Again. So,